Hi, my name is Jason Eades. I'm a PhD student in biomedical engineering at Texas A&M University, and I'm using microphysiological models of human vasculature to study the conditions under which and the mechanisms by which SARS-CoV-2 leads to vascular dysfunction and thrombosis. There is now substantial clinical evidence linking COVID-19 to an unprecedented incidence rate of thrombotic or blood clotting disorders. However, the mechanisms by which these viral infections cause these thrombotic disorders remain unknown. Historically, animal testing has been used to do this sort of research, but in addition to the substantial ethical concerns associated with this practice, these research models do not accurately mimic human vascular physiology and thrombotic disease. Our lab has developed microphysiological systems, or organ-on-chip models, that incorporate human cells into vascular tissues and microenvironments mimicking those found in the body, but outside of the body. I am now using these models to introduce the virus and viral components to study the mechanisms of COVID-19 induced thrombosis. Additionally, I have developed a unique containment system by which these model systems can be implemented in BSL-3 hazardous containment facilities. Using this approach, we are now learning valuable information about the contributors to the onset of thrombotic disorders. Ultimately, my goal is to use this information to support researchers and clinicians' efforts to understand thrombosis and develop effective treatments for it. Furthermore, I hope that this work will motivate researchers to use advanced in vitro models for continued research into human vascular pathologies and drug development.